Thank you for the opportunity to speak to you guys, and thanks to the SJS community for giving me this opportunity. I believe we're all learning something from this. I did learn a lot from Isaac's presentation, so thank you. I'm just going to try and share my screen. I'm not sure if I'm able to. Am I? Or
yeah.
Okay. I'm not sure if you can see my screen now.
Yes, we can.
Okay. So my name is Emanuela, as you previously mentioned, and I currently work for PerkinElmer as a data analyst. You can say working in the testing and tracing of COVID-19. So today I'll be talking to you about the background of coronavirus, and I'm sure you know a lot about this. It's been all over the news, so there's a lot of information out there, but I'm going to try and break it down for everyone to understand. So, what are coronaviruses? And there's actually quite a bit that Isaac touched on. So I was like, oh, don't say he's on that a bit. So, I believe you have an idea about it already. So what are coronaviruses? So SARS-CoV-2, as we may know it, belongs to a family of single-stranded RNA viruses, which are also called coronaviridae. Which is a a common type of virus which affects mammals, birds, and reptiles. In humans, it commonly gives rise to mild infections like the common cold, and also it accounts for 30% of upper respiratory tract infections in adults. So it can also give rise to more serious infections like enteric and neurological diseases. And the incubation period as of now is not really certain of the incubation period, but the United States Centers for Disease Control and Prevention states that symptoms may appear in as few as 2 days, or it can be as long as 14 days after exposure. And Chinese researchers have indicated that SARS-CoV-2 may be infectious during its incubation period. So as Isaac previously mentioned, there's been already some previous types of coronaviruses, and that's the Middle East Respiratory Syndrome, which started in Saudi Arabia in September 2012, and it affected approximately 2,500 people, and it gave rise to over 850 deaths. There was also the severe acute respiratory syndrome, which started in southern China, which was in 2003, I believe, and it affected over 8,000 people and led to approximately 800 deaths. Okay. So where did it all begin? So as Isaac previously said, it began in Wuhan, China, in December 2019, after it was known that there was a pneumonia, case going on around, an outbreak of pneumonia. And experts say that SARS-CoV-2 originated in bats. I don't know how true that is, but it's kind of what people are speculating. And also it's believed that it made the jump to humans at one of open-air wet markets where customers buy fresh meat, fish, including animals that are killed on the spot. So transmission, this is how the coronavirus is transmitted to human to human. So COVID-19 is caused by the SARS-CoV-2 virus, as previously stated, which spreads between people mainly when an infected person is in close contact with another person. The virus can spread from an infected person's mouth or nose in small liquid particles when they cough, sneeze, speak, sing, or breathe heavily. These liquid particles are different sizes, ranging from larger respiratory droplets to smaller aerosols. Other people can catch COVID-19 when the virus gets into their mouth, nose, or eyes, which is more likely to happen when people are in direct or close contact less than 1 meter apart, which is why we've all been told to, keep our distance and all of that. Also, COVID can spread after infected people sneeze, cough on or touch surfaces or objects, such as tables, doorknobs, devices that you also touch after they have touched. So it's advisable to wipe surfaces frequently. So symptoms of the virus. So there are mild symptoms and severe symptoms, as I stated before. So more common and known to us symptoms are fever, loss of smell or taste, fatigue, shortness of breath, and the dry, consistent cough that we've all been hearing about, and sometimes as well muscle aches and pain. And in severe conditions, you might find that there's a difficulty in walking, some sort of confusion, and also probably going deeper would be like kidney failure and high fever. And yeah, so that's your symptoms for COVID-19. And in preventing the virus, in preventing the spread of the virus, we've been advised to stay at least 6 feet, which is approximately 2 meters away from people, distance ourselves and not get too close. Also cover our mouths when coughing or sneezing, and sneezing into tissues and disposing of them straight after. Or also you can cough into the elbow if there's no disposable tissue readily available. And also when in public places, it's advised to wear face masks or any sort of face covering, and also not to touch the eyes, nose and mouth with dirty or unwashed hands, and also to wipe the surfaces and to wash our hands as regularly as possible for about 20 seconds, and also to stay at home if we believe that we have symptoms. So we'll move on to the diagnostics, which I work in this area. So how we test for COVID. So I'm gonna talk about 2 different molecular diagnostic methods, you can say. So the first one is more testing for a patient who has a current or active case of the virus using polymerase chain reaction, which is PCR. And also the second one that I'll be talking about, not going deep into it, is the antibody testing with ELISA. So to test for COVID, to test for COVID in a patient, first of all, a swab is taken inside of the patient's nose or at the back of their throat, and this test sample is taken to the laboratory. So for example, where I work, there's different labs, doing different things. So in the first lab, you would say, or the sample reception lab, we would receive all these samples, all these swabs coming in, and the virus, you can say, is deactivated in a heated oven. And then it is then sent over to the RNA extraction lab, where the RNA of the virus is extracted and purified. And an enzyme reverse transcriptase converts the RNA into DNA, because PCR can only amplify DNA and not RNA. And so the DNA is mixed with primers, sections of DNA designed to bind to characteristic part of the virus DNA, and then it is repeatedly heated and cooled in the thermocycler to give rise to the fluorescence that you see. So in the mixture as well, there's a fluorescent dye molecule which binds to the virus DNA as it is copied. So for example, where I work, different I would say hospitals or companies target different parts of the genome of the virus. And so where I work, we look at the N protein or N gene, which is involved in the packaging of the RNA genome. And we also look to amplify the open reading frame or gene, which is involved in transcription and replication of viral RNAs. So because there are fluorescent dyes which are specific to these parts of the genome of the COVID-19, once it's going through the PCR and these genetic parts are present in the patient sample, it will amplify it above threshold that has been set by the according to manufacturer guidelines. And so if there's if the patient is positive, for example, it will amplify above that threshold, making us aware that this patient sample is positive. And then my job is then to analyze the data that has been given or the graphs presented, and then to using my own discretion or knowledge to mark it as positive or negative. Having said that, there's a few issues with this method of testing. It's not perfect, even though it's quite specific, and it can tell you whether you're COVID or not straight quite fairly quickly, maybe not all that quickly. There's a few issues with it in terms that there's a high demand in the reagents, and therefore there's delayed testing in some countries because reagents are not in huge quantities. And also it can be time consuming because of the step of reverse transcriptase where RNA is converted to DNA. And also it can give rise to false positive and negative results if the sample is contaminated. So when we come to the antibody testing, this form of testing tells the patient whether they've previously had COVID-19. And so it is based on the fact that after 2 days, your spleen produces the antibody IgM, which attacks coronavirus, and after 9 to 11 days, there's more specific antibodies produced, the IgG. And therefore, taking a blood sample from a patient and performing the enzyme-linked immunosorbent assay will determine whether there are these antibodies present, and therefore would mean that the patient had previously been infected with coronavirus. So some pros to this testing method is that it's low cost and it can be used to test large populations. The cons to it you can say that it can't tell you whether you have COVID-19 presently. And treatment methods. So as we all know, there's been a a quick rush. Everyone is trying to find a vaccine for COVID, and there's been more than 300 COVID-19 vaccine candidates currently in development, and 40 are in clinical trials. And in the UK, we've pre-ordered 357 million doses of different potential COVID-19 vaccines from several manufacturers. So part of these ones that UK has ordered, part of it, some of them are two adenoviral vector vaccines, which is more commonly known as AstraZeneca or the Oxford vaccine, and the Janssen vaccine. Also, the UK has sought to reach out for the two mRNA vaccines, the Pfizer and the Moderna vaccines, and also one inactivated whole virus vaccine, the Valvina vaccine. I might be pronouncing these wrong, forgive me. Forgive me if I am, but yeah. And then also the two protein adjuvant vaccines, Sanofi and Novavax, are currently in the run. So 3 of the vaccines being backed by the UK. Not all of these are in use at the moment, but 3 of them are the Pfizer, Moderna, and AstraZeneca, which released positive results from phase 3 trials in November 2020. And the Pfizer vaccine was approved by the Medicines and Healthcare Products Regulatory Agency, the MHRA, on 2nd December 2020. And the AstraZeneca vaccine was authorized for use by the MHRA on 30th December 2020. And the Moderna vaccine was authorized on the 8th of January 2021. So these have been found quite quick, because normally it takes years to develop a vaccine. So it shows that our scientists and researchers have been, working really hard. So COVID-19 and BAME, so where it affects us is where we're trying to learn more about. So I'm gonna play a YouTube video just to give you an insight as to what's happening in the BAME community with regards to COVID-19. Okay.
Factors relating to poverty are not going to be those that are killing doctors, and yet we're seeing they're disproportionately affected. Something like the first 12, maybe more, were BAME. The statistics are telling us that ethnic minorities in the UK are more at risk. The first that came out were that around a third of patients in intensive care units are BAME, Black, Asian and minority ethnic. And this compares to them being about 14% of the population of England and Wales. It's really been notable amongst health workers as well.
NHS medics are dying, and Dr. Alpha Sadhu is one.
Amjad El-Harani was an ear, nose and throat specialist.
Edmund Adedeji died on Wednesday, the 8th of April.
Mary Agyapong, said to be a highly valued and loved nurse.
Organ transplant surgeon Dr. Adel Altaya, volunteering for NHS work in the Midlands.
Lynette Cruz, Melo Jean Ballesteros, Cheryl Williams, Oscar King Jr.,
Faiz Ayash, a Syrian British medical doctor.
And whilst there are more BAME people in the NHS compared to the wider population, it's still only 20% of staff. And I think it's...
Oh, hello, can you see the video? Because I think I've been told you can't see it.
No, we can't see the video at the moment. We can only hear it.
Oh, okay, sorry about that. Can you still see my screen?
Yes.
44% of medical staff, so it's still really disproportionate. The latest figures I've seen are for all the healthcare workers who have died, something like 63% are BAME. We don't know for certain what is the reason, but there are a number of possible explanations, and they include existing health inequalities. For example, BAME people are more likely to suffer from heart disease, kidney disease, diabetes, and hypertension. These are all illnesses which increase your risk of dying from COVID if you get it. There are several reasons why BAME people are overrepresented in terms of these diseases. One is poverty. That affects your susceptibility to certain diseases, and BAME people tend to be overrepresented in people on lower incomes in the UK. There are also genetic factors. So, for example, South Asian people tend to store more fat around their abdomen and other parts of the body around vital organs, and that makes them more susceptible to these diseases as well. Another factor that some people have cited as a possible explanation is vitamin D. According to some studies, vitamin D helps boost the immune system. The problem is that BAME people absorb less vitamin D through the sun than white people. But it's important to point out that there has been no proof that vitamin D protects against COVID-19. As well as being overrepresented amongst healthcare staff, they may also be overrepresented amongst other key workers. BAME people tend to be in lower paid work with more uncertain contractual arrangements. For example, many couriers are BAME, and couriers are one of the professions that are still working throughout COVID. Similarly, bus drivers, and I think 25% of TfL staff are from a BAME background. Other social factors are that BAME people are more likely to live in overcrowded accommodation. For example, 30% of British Bangladeshis live in overcrowded housing. Now, this compares to just 2% of the white British population. And with COVID, the more people you're exposed to, the more your chances of contracting the illness. When we see the amount of doctors that are dying who are from a BAME background, issues of poverty and education are not really relevant to them, which suggests other factors might be at work. One of the key things in the UK that hasn't been collected is the BAME data for actual cases. We just don't know how many of the people who have tested positive for coronavirus are BAME. It's important we understand why minorities are more at risk, because there may be very real measures that can be taken to lessen their risk. And until we know just what's causing it, we just don't know what those measures can be.
Okay, so just continuing with the presentation. I hope you can still see. Okay. Can you hear me?
Yes, we can.
Yeah. Okay, so from that video, we noticed that there are quite a few factors that affects that are causing the disproportionate infection rate of COVID in BAME, and this includes inadequate housing, where there's overpopulation in our houses, and we tend to gather in communities, and also there's more people, there's more BAME working in high-risk jobs, as I mentioned, and then vitamin D deficiency, although this is not certain, there's no scientific proof for this, and also discrimination in the health system, where priority is given to certain people of ethnic and of other ethnic backgrounds, and also there are different health conditions and risks that make BAME more prone to coronavirus. So some statistics for you. The virus has now spread to over 200 countries and territories across the globe after it was characterized as a pandemic by the World Health Organization on 11th March 2020. And as of 20th January 2021, there were 94,963,847 laboratory confirmed cases. And of these, there were 2,029,084 reported deaths to WHO. And as of 18th January 2021, there have been 3,433,494 confirmed cases of the virus in the UK, and of those 89,860 deaths, which is quite sad, really. The fight for vaccines and the vaccines working continues. And so looking forward, there's still emerging SARS-CoV-2 variants, which doesn't help the situation really. In the UK, there's been a new variant identified known as B117, which emerged with an unusually large number of mutations. This variant has since been detected in numerous countries around the world, including the United States and Canada. And in South Africa, there's another variant known as the B1351, which emerged independently of B117, the UK one, but shares some mutations with the UK variant. Cases attributed to this variant have been detected outside of South Africa. And in Brazil, there's a variant known as P1, which emerged and was identified in 4 travelers from Brazil. So, and from this variant, there has been identified 7 unique mutations, including 3 in the receptor binding domain of the spike protein. So yeah, that concludes my presentation. I hope it was of help, and I would like to thank SJS for giving me the opportunity once again. So thank you for your time.